In this presentation, we'll be going over what chemical reactions are and what enzymes are and how they interact with each other to function. Chemical reactions involve the breaking and forming of different types of bonds. In order for the bonds to break, energy is needed for the reaction. When the bonds break, energy is released. Any change in energy is usually in the form of heat. Your first keyword is enzyme. Chemical reactions have two parts, a reactant and a product. The reactant is the element or compound that goes into the reaction. For example, move me over here. For example, in this picture provided over here, the hydrogen is a reactant and the oxygen is another reactant in this chemical reaction because they are going into the reaction. They are before the arrow. The second part is the product, which is the element or compound that is present after the reaction is complete. This will usually be after the arrow. So over here, the product of this is two water molecules that were made from the reactants of hydrogen and oxygen. Always think about the product is what you're going to want to be producing at the end of the reaction. There are two types of chemical reactions. The first one is exothermic, which is a reaction in which heat is given off. I always think of it as exiting, exothermic, EXO, exiting. So heat is going away from the reaction. And then endothermic is a reaction in which heat is taken in. So it has an N in the name, endothermic, so it's going into the reaction. Your second keyword is denature. D-E-N-A-T-U-R-E. We'll be going over that later in the presentation. Endothermic reactions first is a change that requires or absorbs heat. So that's example of that would be photosynthesis as an example. That is when plants take in heat and then they make glucose, which is a sugar. So that is a reaction that requires heat to go into the reaction, or it absorbs the heat. So over here, this little box, you can see that the heat is going into the box and the temperature is going up. A exothermic reaction is a change that releases heat, so it's exiting the reaction. So an example of that would be burning fossil fuels. So during combustion, heat is getting let off into the air. And you can see in this little yellow box here that the arrows are pointing out. So it's going off of the reaction. Every reaction needs energy to get started. So this type of energy is called activation energy. So you always need activation energy to start a reaction. In this chart, the amount of energy needed is on the y-axis, and then the time it takes for the reaction to occur is on the x-axis. You can see that the reaction needs about this much up to the top of the hill here. It needs about that much activation energy. So these arrows right here are the example to get started for the reaction. Once it gets to the top of the hill, then the reaction can take place, and then the products are released at the end of the reaction and are created. So from the amount that the reaction starts till the top of the hump, that is called activation energy. That's what's needed to get started for the reaction. These are examples, I'm going to put me up here. These are examples of exothermic and endothermic reactions. So over here, the exothermic one, this is saying that the reactants are starting up here. It only needs this much activation energy to get started, and then the products are all the way down here. So it is actually releasing this much energy into the atmosphere as heat. It's exiting the reaction. Over here on the endothermic reaction side, it takes this much activation energy to go in, and then the products are released up here, and it's actually absorbing the rest of that energy that went in over here. So it's going into the reaction. All right, catalysts. Catalysts help chemical reactions happen faster. This is done by decreasing the amount of activation energy that is required for the reaction to get started. If you look at this chart here, the top line is lighter, and that is showing that the reaction without a catalyst. So that's without the catalyst that helps make the reaction happen faster. So that's it originally without a catalyst. It's showing that it takes longer for the reaction to happen. So it takes longer for it to get to the top of the hill. It does nothing making it go faster. So it takes longer and it needs more activation energy to get started. While the bottom one is with a catalyst, you can see that it doesn't take as long to get up the hill, and it actually is shorter because of that. All right, your third keyword is in, I-N. All right, so enzymes are substances that are produced by living organisms, and they act as catalysts. So that's what's going to take place. Acting as catalysts, they're going to make the reaction speed up. So the meaning of catalyst means to speed up. So in this case, enzymes are speeding up reactions and producing an end product quicker. An enzyme is a type of protein, and these proteins act as catalysts, and they will speed up all your reactions. Alright, so proteins can be enzymes. I'll move me up here. So here's an example of the process and how enzymes work. So up here you can see this green shape, which is actually an enzyme. So that green one is the enzyme. This yellow one over here is the substrate. So enzyme green, yellow substrate. The substrate will actually bind to the enzyme in this little site that's called an active site here. So it will fit in that enzyme's site. When they bind together like this, this is called a enzyme substrate complex. When they're together like this, they form that type of complex during the reaction. When the reaction is complete, okay, the enzyme will go back over here. You can see that the substrate turned into products. So that's the end of the reaction. The substrate changes and it makes the products that the reaction wants to make. But the enzyme comes out unchanged and it can be ready to be used again. So it should look exactly like it did in the beginning. The only thing that's changing is the substrate that is going in and producing those products. So the enzyme can be reused if it's unchanged. And your fourth keyword is acidic. A-C-I-D-I-C. So only certain enzymes bind with certain substrates. So think of it as a kind of a lock and key method, how only certain keys can fit in certain locks. So that's the same that goes for an enzyme and a substrate. So the key acts as the substrate and will only fit in the certain lock because it's only a certain shape. All right, at the end of the reaction, okay, so at the end of the reaction over here, the enzyme is unchanged and it should be able to be reused again because it looks the same as it started over here on this side and as it does at the end over here. So over here you can see that the substrate fits exactly in that active site of the enzyme. It goes through its reaction and the substrate changes to the products and gets released, but the enzyme stays exactly the same. Your fifth keyword is and, A-N-D. Alright, there are ways that an enzyme will stop working and not be effective anymore. The two factors that cause this are change in pH, which we talked about, acidic and basic, and also change in temperature. So if the conditions are too acidic, so remember that's 6 or below on the scale. So if it's too acidic, it will not work anymore, or if the temperature is too hot. So if the temperature gets too hot or the pH is too low, that means that the enzyme will denature. That means to change shape. So the enzyme is not going to be the same shape as it was before. You're going to need to know that word, denature. This will cause the enzyme's active site to change shape, so it will not bind to that substrate anymore. Therefore, the reaction will not occur at the time that it's needed. So over here in this picture, you will see that the enzyme usually looked like this. Okay, and the substrate could bind right into its active site, but the heat, it was too hot, so the enzyme flattened out like this, and that means that the substrate will not be able to fit in there. So, for example, in our bodies need to maintain a certain body temperature. If our body temperature gets too hot, meaning that we have a fever, certain enzymes in our body will not be able to catalyze our reactions that we need to survive. This eventually will cause our bodies to shut down and causing us to die. That's why it's very important to control or get rid of a fever quickly, even though it is our body's way of responding to certain aspects. Okay? So your sixth keyword is hot, H-O-T. Alright, so these pictures show an enzyme that can be affected by the change in temperature or pH. The active site will change shape, meaning that this substrate will not be able to bind with the enzyme anymore. So here the enzyme started out looking like this. The substrates could bind to it, but over here, if it gets too hot or the pH changes, that means that they will not be able to fit in the enzyme anymore because it denatured or changed shape. So for example, this is why our bodies need to maintain homeostasis. Homeostasis means to keep at a constant temperature or pH, so our body regulates our pH and temperature. If our body gets too hot, meaning we have a fever, the enzymes will denature and will not be able to catalyze the reactions, and eventually we would die. You will usually see enzymes ending in the names. They will end in ASE, and that's ACE. The beginning of their names will tell you what the enzyme does. So for example, protease, so the beginning, okay, sounds like protein, and then ACE at the end, it means it's an enzyme. So Protease means to break down proteins. The second one, DNA polymerase, which we will talk about, it polymerizes and means that it joins to make DNA. Joins polymers to make DNA. And then the last one, ATP synthase, means that it synthesizes or makes DNA. So any enzyme ends in ASE, pronounced ACE. And your seventh and final key word is conditions. 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 If you have any questions, please ask your teacher. Make sure you have your notes filled in. And good job.